Now I want to talk about how you can connect different regions of your brain, such as your cortex, prefrontal cortex, and hippocampus. There are other important parts that are essential in what we do because they are interconnected, such as striatum that is inside in your basal ganglia, which is for decision making, reinforcement learning. Paradigm in machine learning is very related to this basal ganglia, but the structure of striatum is still unknown. In this lecture, I will focus on the connection between prefrontal cortex, because prefrontal cortex is like working memory. It's very fast, but it forgets, so it gives information to the cortex, different parts of cerebral cortex, and of course this is part of cortex. But hippocampus is responsible for consolidation. When you go to sleep, it tries to index everything it has learned through the day, and hippocampus has different parts, C1 through C4, and many different parts, but it does pattern completion, it does pattern separation, and many different tasks. And so let's focus on modeling this part, which is also connected to part 3, which is in the next lecture, of course. And I want to explain this beautiful article 2014 from a very great professor, Anders Lassner, in his laboratory. He has many great students and colleagues, and his work is very important for the future of biological neural networks, and also its connection with artificial neural networks. Because biological neural networks are millions of times more efficient than these artificial neural networks that we are familiar with. And what is episodic memory consolidation? We propose a functional neural network implementation of these ideas and further suggest an extended 3-stage framework that includes prefrontal cortex. It bridges the temporal chasm between working memory percept on the scale of seconds, which is very fast, and consolidated long-term memory on the scale of weeks or months. So if you don't sleep well, then you cannot understand and learn efficiently. And so we show that our 3-stage model can autonomously produce the necessary stochastic reactivation dynamics for successful episodic memory consolidation. It is generally accepted that neocortex provides a slow learning substrate for distributed long-term memories. Fast working memory capable of immediate one-shot learning has been localized around the prefrontal cortex. The discourse on declarative memory consolidation has, however, been largely centered around hippocampus and various substructures of wider medial temporal lobe, which store memories on an intermediate time scale and thus are perfectly suited to moderate the consolidation process. So, as I said, hippocampus has many different areas, such as CA1, CA3. Those are for pattern completion and pattern separation. So, I always think about the brain as a microservice architecture. Unlike artificial neural networks, that is like a monolithic architecture, everything is just in one neural network. In your brain, each component does it is in harmony with separation of concerns principle in Java principles, in programming mathematics, in programming principles in general. That each module should only focus on what it does. It should only focus on a very specific job, very specific task. unlike artificial neural networks that we have a module, we call it neural network, and everything has inputs and outputs. But this is monolithic. This doesn't work. This doesn't scale. This doesn't work efficiently. In biological systems, everything is like a microservice architecture if you're familiar with software architecture. I have been a software architect, I've been a microservice architect, and I know it's very similar to what biology is. In biology, everything is a microservice, everything is a module, and there is lots of feedback interconnection between them to regulate these variables. And we don't do, our mathematics is just by doing feedback mechanism. And so our primary sensory cortices, such as auditory cortex, visual cortex, you name it, there are lots of them. They come and they separate. It says, this path says, what pathway? What is it? What pathway? Is it a cat? Is a picture of a cat? What is it? And where is it? For example, in a mouse, they understood that place cells that are part, but everything, you can generalize this idea to everything. Everything you think has correspondence in your brain, a set of neurons that are responsible for that. So parahippocampal cortex is responsible for this where pathway, and entorhinal cortex is very interesting. It combines these information and give it to the hippocampus in an appropriate, filtered, modulated way. And there is a dentate gyrus as well. This dentate gyrus is a region in the mammalian brain, human brain, for example, critical for memory encoding with a neuronal architecture. So it encodes our memory. And the periorinal cortex is for object recognition memory, but the parahippocampal cortex is for memory and navigation, and you know those experiments that they have done on mouse. That, for example, where the mouse is, and depending on just one cue, the mouse understands the rest of it. And what is the function of entorhinal cortex? The entorhinal cortex is a region of brain located in the medial temporal lobe, and these are the functions. But I don't have time to go through them. They are very important. Memory formation, spatial navigation, memory integration, time conception. But it's a recent research. Nobody knows if it's correct. Alzheimer's disease. So let's focus on hippocampus. You know, CA1, CA2. It's like a pipeline through your architecture. And CA1 neurons, this one is very interesting, are essential for retrieval or remote episodic memory, and that they are important for autonomic consciousness. And CA3 is important as well. Through its recurrent collateral connections, it's a single attractor enabling fast one-trial association between any spatial location and an object or reward. Of course, the reward mechanism is in striatum, which is in basal ganglia, but it just indexes what should go into striatum. So it has been suggested working memory performance may be aided by hippocampus, especially for relational processing. But more recent studies reveal that working memory performance remains unaffected by hippocampal and even wider lesions if the capacity requirement do not exceed a narrowly defined working memory capacity. Hippocampus may still aid working memory by extending the available capacity. So this is the architecture, how your hippocampus is also gives information to the thalamus. We know thalamus is the gateway of all sensory informations, but it is more than that. We know amygdala is for our emotions, fear, understanding that. So what is challenging? The temporal scales on which working memory, intermediate memory, and long-term memory operate are separated by many orders of magnitude in time. For example, prefrontal cortex is very fast, and after that we have hippocampus, which is intermediate. But there is very long time scales, which is very slow, but it is a part of cerebral cortex, and it's responsible for many reasoning and many things. So hippocampus, prefrontal cortex and cortex, because they are part of cerebral cortex, we use this because cerebral cortex has 6 layers, and each layer has hypercolumns. We call them hypercolumn. For example, this is a hypercolumns, but inside the hypercolumns there are many mini columns. But all of this is just 5 micrometer, for example. But inside each micro column, there are lots of neurons inside that. So there are lots of neurons inside that. But each microcolumn is for different features. For example, color. These attributes that we understand when you say a word, you will see that in your functional MRI, that expensive machine. But the interesting thing is that everything is distributed, everything is sparse, because just one or very few nodes in each mini columns are active, and the rest of them are just 0. So you see, it's a very, very sparse. That's why I'm interested in sparse distributed memory, because many things in your cerebral cortex and also in your brain in general is very sparse. And also, it's distributed in different parts of your brain, hippocampus, prefrontal cortex. You will see in future, I will talk about striatum in your basal ganglia. And so you see everything is distributed. Hippocampus, just a microservice. It focuses on something. Your prefrontal cortex focuses on working memory. Everything has a specific task. It's not a monolithic architecture just like in your artificial neural networks. That's why it's very efficient. And everything is for a reason. So prefrontal cortex used for working memory, and also hippocampal memory, the second population modeling the intermediate term hippocampal memory system, might anatomically involve close by areas of MTL such as perirhinal cortex, and parahippocampal area is 5 times larger and much slower learning with this time constant. This time constant is very important in control systems. We have different blocks in control system. We connect them, and so it's more similar to how brain is in comparison to those naive artificial neural networks, that everything is a monolithic, just inputs and outputs. For example, even they are working on this monolithic architecture and work on continual learning, you will see that still their paradigm is on monolithic architecture, which is not natural. But our brain, why don't we look at the brain? Everything is a microservice. Everything is distributed, and everything is modular. Everything for a different task, for a different purpose. It's distributed. And the third population that we want, and in previous lecture I was talking about population, those Wilson-Kowal model that I told you, DI, the inhibitory, DT is a vector field, the inhibitory and excitatory. So this is a vector field, and this dynamics depends on the fixed points of these dynamical systems. So at perception, at sleep there is no prefrontal cortex. At sleep, prefrontal cortex is dead. There is no consciousness at the meaning because it's very fast. When you open your mind, you're awake, then it was working. But when you're asleep, it doesn't work. But there is a kind of sleep that is in, I forgot the name. There is it is between sleeping and being awake. that maybe that prefrontal cortex is also working in a very sparse way. So what are these columns? These columns and rows that you see, I will explain in the next lecture. But you should know that our cerebral cortex has 6 layers, and this is the gray area. 1, 2, 3, 4, 6 layers. And also they're related to subcortical areas such as thalamus and striatum, but the more 90% of connections are between them, even between columns, even between layers, even inside. So the recurrence is more important than these subcortical, because just 10% or 5% of them are subcortical. So you see that recurrence plays an important role for your intuition, understanding, reasoning, comparison, and different higher order cognitive tasks. So this is the model that I will explain in the next lecture, because you should know who designed that. I know it was in 2001 by the great author in KTH, and his team are still working very successfully. Their work is very important. I don't know why people don't read those articles, because they are very important articles in KTH University. And so you see here, we want to know, as I said, we have different layers, and each one has hypercolumn, and each hypercolumn has mini columns, and inside those mini columns there are neurons, and not only this, but also it has 6 layers. So at each layer we have these columns, and also there is a feedback connection between them. So it's a very complicated structure, but we can model it in a very simple way, at least behaviorally. For example, what if this term becomes 0? Then if you just reverse engineering that, you will see that it's just the probability Bayesian law. Because this is related to this network, Bayesian propagation neural networks. And so I will explain it in the next lecture. But it's very simple. And this is the probability that, for example, you select a special node, which corresponds to a different attribute. For example, you have some nodes, some neurons in for example, in presynaptic, and in postsynaptic we have your target, some neurons. But these neurons are inside those hypercolumns. So at each hypercolumn there are several mini-columns, but at each mini-columns only one thing is selected. So what is the probability that if they are active, then something else is active? What is the probability? And when it converges, we understand that, for example, this node is active or this node is not active. The connection is important. If I is connected to J's, if I's neuron is connected to J's neuron in the postsynaptic area. And of course, so this one becomes input and output, and then we fix these weights, and we go to the next layer. It's very similar to some of the techniques in Boltzmann machine, those deep belief networks, learning of them, which is an alternative to, because we are focused on Hebbian learning, which is a local thing. But in artificial neural networks, everything is backpropagation, which the error backpropagates through the network. And this is not natural. This is not what is happening in your brain. Your brain does everything very beautifully in a local fashion. Everything is local Hebbian learning, just a very simple Hebbian learning. Even in Hebbian learning, genetics and epigenetics is also considered. I will show you why. Even the location of neurons has a different gene expression and then generates a different neurotransmitter, neuromodulator. A different neuromodulator depends on the gene expression. So you see, it's a neurogenesis. You combine those DNA effects, the effects of DNA as well as the neural networks. So that the biological networks, everything is local. For example, the Hebbian law, it's just between the neurons around that, and not the neurons that are distant about it. It propagates naturally, but the error does not